Intracellular inclusions in bacteria. So as you can see in this picture, every bacteria, almost every bacteria would have, certain granules that are present inside the cytoplasm, as one can see over here. So apart from the ribosome particles in a bacterial cell, you have small, big granules, which are called as inclusion bodies as well in bacteria. And these inclusion bodies are known to help bacteria to survive under very difficult conditions. So let us look at the learning outcome of this session. So inclusions are discrete structures present in the cytoplasm or in the periplasmic space. They are formed due to the cell's metabolic expression. Depending on the type of inclusions, they can have different functions. And these inclusions may or may not have membranes. Inclusions as metabolic organelles. So we will first take up two examples of organelles. So, and you call them as organelles because they begin to carry out certain very specific functions. So the first metabolic organelle that is being considered is carboxysomes. So within, especially bacteria like cyanobacteria and other chemolithophotoautotrophic bacteria, those that are able to synthesize their own food by harvesting light, so photoautotrophs basically, they are observed to have these inclusion bodies which are called as carboxysomes. A higher resolution of these carboxysomes shows that there are basically two different types of carboxysomes, alpha carboxysomes and the beta carboxysomes. And these carboxysomes are generally polyhedral. They are three-dimensional polyhedral structures. And within these polyhedral structures are present rubisco, that is ribulose bis phosphate carboxylase or oxygenase, which means this is one of the major enzymes that is involved in carbon dioxide fixation, and it can act as a carboxylase or it can act as a oxygenase depending on the concentration of carbon dioxide and oxygen. So thereby these polyhedral structures, which are carboxysomes, contain the rubisco, and each of these polyhedral shells are covered by a thick shell which is made up of carboxysome shell proteins, that is CSSO proteins. There are various different types of these proteins that form the thick shell, but this shell is formed in such a way that there are pores existing in this shell. And because of the presence of the pores, substrates can enter the carboxysomes, and the substrates mainly are in the form of bicarbonate. So when bicarbonate enters the carboxysomes, the rubisco is able to use up the carbon dioxide in the bicarbonate and fix it to give rise to a product that can be again released. Now, because the thick shells are made up of specific proteins, many of these proteins do not allow easy gas exchange, and so therefore there is specific gas exchange that happens, not allowing oxygen to enter. Because say for example oxygen is not allowed to enter, then the oxygenase activity of the rubisco does not happen or does not take place, and therefore it will only function to carry out carbon dioxide. So basically the polyhedral carboxysomes are organelles in which carbon dioxide is being fixed. So this is what their major role is. Now, interestingly, what is observed therefore is there is a continuous inflow of the bicarbonate ions, and when they flow into the carboxysomes, because it's a closed structure, you would have a gradual buildup of the HCO3, and because HCO3 minus, that is bicarbonate ion, and because there is a slow increase in the concentration of the bicarbonate, it comes nearer to, or it is in the proximity of the active site of rubisco, and thereby that carbon dioxide can then be fixed. As mentioned earlier, it regulates gas exchange, and so it ensures that there is minimum oxygen present within the carboxysomes. So this is a metabolic organelle that is present in many photoautotrophs. Now we come to the second metabolic organelle as an example, and that is chlorosomes. As the name suggests, it has chlorophyll. And interestingly, these chlorosomes are formed from the plasma membrane, that is a cytoplasmic membrane itself. You can see that it can stem out from the cytoplasmic membrane. But what is interesting to note is that the membrane that is formed is asymmetric. So you have the outer layer of the membrane, which has several proteins and of course lipids, and the inner layer practically is made up of the bacteriochlorophyll molecules itself. Now, bacteriochlorophyll, because there are many numbers of bacteriochlorophyll present inside the chlorosome, and this chlorosome being able to associate itself with the uh, photosystem 2 or the photosystem of the bacteria. So light is harvested, and that harvested light leads to excitation of the bacteriochlorophyll, which is able to bring electrons to an excited state, which then is transferred to different bacteriochlorophyll molecules, eventually leading to the reaction center. And at the reaction center, then what happens is you have the electrons getting into ferredoxin to form reducing equivalence. So therefore, one can again see that light harvesting is the role of the chlorosomes. And since it has one particular function that is being carried out inside the chlorosomes, you have this also acting as an organelle. So these organelles are highly variable in their length and diameter. That has been observed by various studies, and that is attributed to the fact that the bacteria live under various different conditions, and depending therefore on the functionality of the organism, the light intensity and the environmental conditions, you have not just a lower or higher number of chlorosomes, but also you have their shapes different. So some can be long, some can be short, likewise. Next, we go to inclusions that are associated with motility. So many bacteria have what is called as gas vesicles present in them. As seen in this diagram, the gas vesicles that form initially are just small globules which eventually grow to form what is called as a clinical cylindrical structure. And uh, in many bacteria, 90% of the entire bacteria is actually just the gas vesicles. So the question now arises is, why are gas vesicles needed by the bacteria? So what has been observed is that gas vesicles are majorly present in aquatic microbes, that is bacteria that are present in water, and the presence of the gas vesicles inside the cell enables the cell to float, or rather what happens is it becomes buoyant. And because it becomes buoyant, it is able to move towards an environment of more light and oxygen. So in a way, the presence of oxygen or the amount of oxygen enables the cell to move towards a place where you have more oxygen. So from very highly anaerobic condition, the bacteria can move to microaerophilic conditions, and from microaerophilic they can also go to aerobic conditions. So you can see that the cell has moved from one environment to another environment thanks to the gas vesicles. This is primarily because the gas vesicles are able to exchange several different types of gases, including nitrogen. So they are able to take up oxygen, they are able to take up carbon dioxide, they're also able to take up nitrogen. So they're permeable to many gas, and majorly what happens is the cell surface to volume ratio is going to increase. This cylindrical shape of the gas vesicles is attributed to a special protein called GVPA. This protein itself has a coiled structure, and when these coiled structures come together, they form what is called as a rib cage structure. And therefore within this, so it's creating a mesh kind of thing, and through this mesh you can have the oxygen or the nitrogen or any other gas to permeate through. So that is how the structure of the gas vesicles are formed. Then we come to another inclusion body that is associated with motility, and they are magnetosomes. So magnetosomes are found in magnetotactic bacteria or non-magnetotactic bacteria. Primarily, magnetosomes are nothing again but small granules or small bodies that are made up of magnetite, or they are made up of gregate nanocrystals, and they are present within a membrane. So as you can see, these are membranous structures, right? And there are several proteins that are present on these membranous structures that enable the ion to be taken up, ions to be taken up, but in various different forms. So you could have them take up primarily gregate or the magnetite. Also, what is observed is that magnetosomes in bacteria can either exist dispersed or they can align themselves in a particular orientation. Now, this aligning themselves in a particular orientation is again thanks to a set of proteins. How does this alignment help? Well, every magnetosome will act as a single magnetic domain, and so it will, the aligning of them in a particular orientation is dependent on the south pole and the north pole, right? The two poles of a magnet. So they have distinct dipoles present, and each of them will arrange themselves in that dipole order, and based on that, what happens therefore is the bacteria can move towards a magnetic field. Okay, so the cell is aligned along a magnetic field, and because it is aligning along a magnetic field, you can have groups of bacteria moving based on the magnetic field, and that is what is called as, the phenomenon is what is called as magnetotaxis. So a bacteria is attracted towards a magnetic field. Now, because of this special characteristic, many of these magnetosomes have seen applications uh, in several areas. For example, as drug delivery system, or they are used in bioassays, etc. So they have an applicability also because of this special feature. Of course, the synthesis of these magnetosomes depend on several enzymes. So you can see there are lots of proteins present, and many of these proteins act as enzyme that enable the magnetite or the gregate to be taken into the magnetosomes. Inclusions associated with energy reserves. So storage granules are present mostly in all bacteria. And some of the very common storage granules are what we are going to discuss. Glycogen granules. Glycogen, all of you know, is a polymer of glucose. So it serves as an energy or carbon reserve, and it forms when there is a limiting nutrient. So what is observed is that cells that have reached a stationary growth phase, and on reaching the stationary growth phase, there is excess of carbon, but there is a limiting nutrient. Say, for example, oxygen is limited. Then under such a limited condition, what has been observed is that many bacteria tend to form what is called as a glycogen granules, with the objective that if required, these glycogen granules will be used as reserves under dire straits or under very stressful conditions. Now, interestingly, glycogen granules have also been found to sustain a process of sporulation. So when a bacteria begins to sporulate, as you can see, it begins to sporulate, you will find that there is a lot of accumulation of glycogen granules. What is also interesting to note is that glycogen granules help reduce buoyancy. So if you have a bacteria with a large number of gas vesicles, to counter the buoyancy because of the gas vesicles, the bacteria can form a large number of glycogen granules to bow down the bacteria. So again, to counterbalance the gas vesicles, you can have glycogen granules being formed inside the bacteria. It also seems to help in osmoregulation and stress tolerance. So, in fact, it is observed that many mycobacterium which have these glycogen granules are able to resist or survive in cold conditions, and they can also survive desiccation. The next type of storage granules is lipid granules. And there are several different types of lipid granules, of which polyhydroxyalkanoate granules, that is PHA granules, are abundantly found. And of these granules, again, PHB granules are most common, that is polyhydroxybutyrate granules. Again, because they are lipids, their bonds on breaking can be used, or rather they can be broken down to form acetyl-CoA, and this acetyl-CoA in turn can be used for energy production, and so therefore they serve as energy or carbon reserve. And most PHB granules are supramolecular complexes. So you will observe that all the PHB is present inside. So there is a hydrophobic condition present inside, because of which the fat resides within, and it is surrounded by a layer of proteins. And these layer of proteins, for example, there are phasins, there are different types of phasins that are present, and these phasins are forming the outer shell of such PHB granules. You also have what is called as a triacyl glycerol and wax ester granules. So the triacyl glycerol and the wax ester granules again serve as an energy carbon reserve, and they have been found most commonly in actinomycetes. So here you can see these are images where you will find plenty of TAG and wax ester granules present. Likewise, there is something also called as the cyanophycin granules. What is interesting to note is that the cyanophycin granules are made up of, its backbone is basically aspartate, and the side chain of the aspartate is attached to arginine. Therefore, it is basically going to be a polymer of amino acids, that is aspartate and arginine. And this polymer is not synthesized in the ribosome, it is synthesized other than in ribosome. So although you have these amino acids coming together to form a polymer, so we all know that whenever you have a polymer of amino acids, you relate it to being formed in the ribosome. However, these are structures that are not formed in the ribosomes. And also another interesting fact is that these granules do not have membranes. So they are inclusions, yes, but they are membraneless inclusions. And they also serve as energy, carbon, and also nitrogen source, because arginine is rich in nitrogen. So therefore, it serves as either energy, carbon, or nitrogen reserves. Now, both the TAG and the wax ester granules and the cyanophycin granules again are formed by bacteria when there is excess of the monomer of the polymer present, but there is a limiting nutrient. So it is formed when the bacteria is moving towards a stressful condition, or when it is reaching the stationary growth phase. So you know that when a bacteria reaches stationary growth phase, there is some limiting nutrient. So both these are also formed under such conditions. One also has what is called as polyphosphate or volutin granules. These are also called as metachromatic granules based on metachromatic staining. The process that is used to stain these volutin granules gives it the name metachromatic granules. The polymer, interestingly, is a linear chain of inorganic phosphate, and these inorganic phosphates are linked to each other by phosphoanhydride bonds. And it is synthesized more again in the stationary growth phase. So you can see that again the bacteria, when it reaches a condition where there are limiting nutrients, then you have these polyphosphate granules formed. And apart from being a store of inorganic phosphate, it also is able to be a replacement for ATP. So therefore the phosphoanhydride bonds can be broken to give rise to energy. So one can see that when stained with DAPI, that is a fluorescent dye, one gets, one can see in the bacterial cells these metachromatic granules formed. One very easy source of metachromatic granules is curd. When curd becomes highly sour, okay, what what is observed is lactobacillus slowly, gradually begins to form volatile granules. And so when you do metachromatic staining, you will find lot of volatile granules present in the lactobacillus. So sulfur globules are of course present in bacteria that are growing in sulfur rich environment. So for example, green sulfur bacteria or purple sulfur bacteria. And in these inclusions, you have sulfides being taken up by the protein shell. So you have a membrane present, and there are several proteins present in the membrane that are able to selectively take up sulfides, thiosulfates, polysulfides. And these are now a store of reducing power. So when the bacteria has to survive under anaerobic conditions where there is minimal oxygen present or hardly any oxygen present, then these sulfides are able to be the electron donors, and thereby these reducing powers can be used by the bacteria to get energy. So again, therefore, the sulfur globules are present in all bacteria that are present in sulfur-rich environment. Now, we go to looking at cyanobacteria as an example where you have many inclusions present. Now, cyanobacteria generally exist in chains. So you have several cyanobacteria aligning themselves in chains, and under specific conditions, many of the cyanobacteria differentiate into forming what is called as heterocysts, and these heterocysts are able to fix nitrogen. But around the heterocyst, what is also found is some of the cyanobacteria are able to differentiate into what is called as cyanobacterial akinete. This is equivalent to being a kind of spore. So therefore, what happens is that the cyanobacterium akinete, when it becomes or is maturing to form the spore, okay, you have basically several different types of inclusions formed inside. So you will have lipid droplets, you will have the carboxysomes, you will have the polyphosphate granules, you will have the cyanophycin granules, and therefore all of these will help the cyanobacterial akinete to be present, to be surviving in a condition where it cannot multiply. So this, as a hypnospore, will remain for a long period of time, and it is able to survive for a long period of time because of the reserves that are present inside it. So to conclude, inclusion bodies are found in many bacteria, especially as carbon and energy reserves, and are formed either during the stationary growth phase or specific environmental conditions or limiting nutrient conditions. Several inclusions are highly specific in their functions, such as carbon dioxide fixation by carboxysomes, magnetotaxis by magnetosomes, sustenance of sporulation also by glycogen granules, etc. Most inclusions enable the bacteria to tolerate stress conditions such as cold, desiccation, anaerobic conditions, etc. Due to their specific structural attributes, there have been many applications of these inclusions in research and industries. Thus, bacterial inclusions are discrete structures that accumulate or are formed under specific nutrient imbalance conditions, functioning as metabolic reserves, cell positioners or cell motility, and as a stress tolerant component. Thank you.